Hi everyone, I'm Les Loew. I'm going to be talking to you today about reaction kinetics and diffusion. This first segment of about 15 minutes will be a repeat of the first 15 minutes of the talk that I gave last week at the Computational Cell Biology Workshop, but somehow it got corrupted, so it's going to be spliced into the original talk. And there won't be any questions, obviously, for this first 15 minutes. But I will remember them and try to answer them sort of as part of the presentation. So without further ado, let me share the screen. And these are the objectives. We're going to be talking about dynamics of individual molecules, the laws of diffusion, stochastic and deterministic treatments of kinetics, and more complex kinetic systems, steady state mass action versus more complex rate expressions, and then understanding how all of these processes, diffusion, kinetics, membrane transport, are coupled within the spatial environment of a cell. So let me get the laser pointer up. And let's continue. So let's start with diffusion. This is a single molecule, as you can see, an animation of a single molecule that's bouncing around, bouncing off solvent molecules in solution, and moving in a random way. When we see this kind of random movement or any sort of random unpredictable behavior, the term that's used is stochasticity. This is a stochastic process because we can't predict exactly what's going to happen, but we can give it some sort of probabilistic prediction of what's going to happen. So we don't know when it's going to cross that barrier that opened, but we know that eventually it probably will. It probably will find its way to the other side of the barrier. If we have many molecules, or if we follow many individual trajectories of this one molecule, we can define a probability that the molecule will move a certain distance. And it turns out that that probability, the probability that the molecule will move a distance delta x, is given by is proportional to the square root of time. And the proportionality constant is called the diffusion coefficient. So here's another way of writing the same thing. The mean of the square displacement is proportional to time, where the proportionality constant for 3D at least is 6 times this diffusion coefficient. We can also think about what happens when you have more than one molecule. And so here are the corresponding animations for one molecule, a small number of molecules, and a large number of molecules, which can be described continuously as a concentration. So initially we have this barrier. We're going to break the barrier and see what happens. And as you can see, when you have more than one molecule, then probably eventually you'll have a similar density on the other side of the barrier as you have on the initial side of the barrier, on the left side of the barrier. For concentrations, you can describe this as a continuous process instead of as a stochastic process where you have to deal with probabilities. We describe this deterministically using differential equations. And the differential equation that tells you how many molecules will cross that barrier as a function of time is Fick's first law. It's given by this equation. J is the number of molecules crossing a unit area per time, is proportional to the diffusion coefficient times the concentration gradient. The larger the concentration gradient, which is obviously largest at time 0 before the barrier, right after the barrier breaks, the faster the molecule is going to move across. Once that number of molecules sort of equalizes out, then there might be an equal number of molecules moving in either direction, and the flux becomes 0. So at equilibrium, when there is the same number of molecules moving in both directions, the flux is 0, and the concentration everywhere is equal. So let's look at this proportionality constant D and think about it for a minute. This, in fact, wasn't the answer to one of the questions. Somebody wanted to know what controls D. So D is proportional to 1 over the diameter of the molecule, say a protein molecule. And since it's 1 over the diameter, that would be proportional to 1 over the cube root of the volume, since volume is proportional to the diameter cubed. Or if it's a square or a cube, it's proportional to the cube of one side. If you think about it, the molecular weight, of course, is kind of proportional to the volume of the molecule, if we're dealing with a protein, for example. So the diffusion coefficient is proportional to 1 over the cube root of the molecular weight. This proportionality holds for a sphere, but that's a good enough approximation for many globular proteins. So this means that doubling the molecular weight really doesn't change the diffusion coefficient by very much, just by 20%, because 1 over the cube root of 2 is just 0.8. And therefore, D is not that sensitive to the size of a protein. D is also proportional to the viscosity. So D inside the cytosol is going to be slower for the same molecule than D inside a simple water solution in vitro, usually by about a factor of 3. The viscosity of cytoplasm is about a factor of 3 greater than the viscosity of water. And what's even more important is for membrane molecules. The viscosity of membrane is sort of like WD-40. And so the diffusion coefficient in a membrane can be as much as 100 times slower for the same size molecule than the diffusion coefficient in water. So that just gives you a rule of thumb. So viscosity is actually very important. And diffusion is also proportional to temperature. The faster the molecules are bouncing around with greater kinetic energy, the faster the diffusion coefficient. So you can put all those together and say D is proportional to temperature, but inversely proportional to viscosity and the cube root of the molecular weight. And the units of diffusion are distance square over time. So we've talked about Fick's first law, where the number of molecules crossing a unit area per second are given by this diffusion coefficient times the concentration gradient. Fick's second law describes how concentrations change, how concentrations change as a function of diffusion, how stuff accumulates if it moves faster in than it moves out. That's Fick's second law in words. So if you look at a little diagram like this, if J1 is greater than J2, then molecules will accumulate in this area and the concentration will go up. You can describe this probabilistically, but we're now in the realm of a deterministic solution, and Fick's second law is given by this equation. The partial derivative of the concentration of this molecule, C, as a function of time, is given by, again, this diffusion coefficient minus the diffusion coefficient times the second partial derivative of the concentration with respect to space. So because there are derivatives involved in both space and time, that's why we're dealing with partial derivatives. If this is a three-dimensional diffusion instead of this one-dimensional diffusion that we're talking about here, then you would just add terms for y and z that would be exactly the same as this term. So that's Fick's second law. And again, it just means that stuff accumulates, the concentration goes up if molecules move in faster than they move out. And you know that they're going to move in faster if there's a bigger concentration gradient on one side of the test area compared to the other. Okay, so we've finished our discussion of diffusion alone. We're going to now go to talk about kinetics. And by the way, if you want to play with diffusion alone in virtual cell, here's a public model that you can play with and see how diffusion changes in different geometries and how the rate at which a molecule can equalize in different geometries as a function of the size of the vessel, etc., as a function of the size of the diffusion coefficient, etc. Reaction kinetics. So let's say, well, let's first think about this diagram that I have here. This is what's called a reaction coordinate diagram. You may remember this from your freshman chemistry. The y-axis is energy, and the x-axis is usually called the reaction coordinate, sort of the trajectory by which A can change into B, how the bonds can change within A to convert itself to B. And let's say that there's enough thermal energy so A can get to B, can get over this barrier, energy barrier, and it does so with a 10% chance for A to jump the barrier within 1 second. So the A molecules have a possibility of jumping to B, but only a 10% chance in 1 second. So if you had 10 A molecules at time 0, how many B's would there be at 1 second? Well, you really can't answer that question because this is probabilistic again. This is stochastic. So we can say, you know, there's a pretty good chance that if this is the rate, if this is the probability, that for 10 molecules, at least one of them might go over to B, but you can't be sure. Or it could even be 3, and maybe even all 10 will go to B in 1 second. Every time you try this experiment, it's going to be a little different. But if you do it often enough, you know that 10% of the molecules, 10% of the time, A will go to B. And so if you had 10 molecules, you know, pretty good chance that one at least will go to B in 1 second. So for a small number of A's, this system is stochastic. But you can also describe this deterministically, continuously, by solving a differential equation. The differential equation is just saying that the derivative of A with respect to time is given by a rate constant times the concentration of A. The more A there is, the more B there will be after a unit amount of time. And that differential equation is just this simple rate expression, which again is something that you should sort of be able to resurrect from freshman chemistry. The change in A with respect to time is given by that rate constant, 0.1 per second, times the concentration of A. It's going to decrease, so it's a negative sign. And you can integrate this very readily, of course, and it gives an exponential. This is the initial concentration of A at time 0, and it will decay exponentially with a time constant of 0.1. Okay. We can plot these things both stochastically and deterministically. These are two plots that were generated by virtual cell. For 10 molecules, you start off with 10 molecules and they start decaying. This is A in blue, and this greenish yellow curve is the corresponding mirror image for the production of B. Every time A changes, it produces a B. And that's for 10 molecules, but if you had the deterministic solution for concentrations, you started, say, with 1 micromolar and solved that differential equation, this would be the deterministic solution, the continuous solution, where A decreases and B increases with these single exponentials. Now, if we, instead of having 10 molecules, went to 1,000 molecules, you'll see that we get these noisy curves, but they're virtually identical now to the deterministic curves that we calculate by solving the differential equation. So this is the stochastic solution for 1,000 molecules, and this is the deterministic solution solving the differential equation. So again, you can look at this V-cell model and play with it yourself, change the number of molecules, change the number of trials, etc., and see how the system behaves, and behaves differently each time you do it stochastically, but obviously is always the same if you solve this deterministically. So we've completed this objective in our list. Let's go to more complex kinetic systems and start off with a bimolecular reaction. This again should be familiar. This is all something that I'm just trying to remind you of from freshman chemistry. A plus B goes to C with a rate constant of K sub F, K forward. And the rate expression here, which is equal to the change in the concentration of C with respect to time, is given by Kf times A times B. That's the bimolecular rate constant. This is called a mass action rate law. And it's the most straightforward, simplest rate law. You could solve this stochastically too. And again, if you want to play with it in virtual cell or in Kapasi, you can do that either way. But we're only going to look at the deterministic solution. So you can, again, integrate this rate expression for a specific set of initial conditions where you know the initial concentrations of A and B. Let's say they're the same at 10 micromolar. Then you can come up with an analytical solution for this rate expression by doing the integration, and you know that the concentration of C is going to be given by this function. And I've just plotted this for two different choices of Kf over here, where Kf is equal to 10 per micromolar per second, and Kf is equal to 1 per micromolar per second. The yellow line is obviously the slower rate. Please take note of the fact that this is in units of per concentration per second. And when you multiply this Kf, which is units of per micromolar per second, times micromolar, the unit for the rate is now micromolar per second. Okay, so it all kind of works out and makes sense. So you can get more complicated with reaction networks. Here's A plus B going to C, but it's also reversible. C can go back to form A and B. And that reverse process is given by a rate constant K minus 1. So you have K1 going forward and K minus 1 going backward. But then C, you know, if you're thinking about biochemistry that occurs inside cells, it never stops with one reaction. C can do something else. And then D is produced, and D might be an enzyme that converts A to something else. So, I mean, there are these reaction networks, and you have to worry about solving the kinetics for complex reaction networks, and that's why we need software. That's why we need COPACI or virtual cell. And let me just show you the version of this same reaction network, A and B going to C, and then C going to D, in a simple reaction network in virtual cell. And here are the rate constants that I've assigned to the forward and reverse rates for this first reaction, the one that involves A and B. Okay, this is called mass action kinetics in both cases. When you have this simple type of relationship. And you can write down the rate equations again for A and the rate equation for C. You can write the rate equation for D. D would just be R2, which is this rate expression, etc. The other thing that constrains this system of equations is conservation of mass. You know that the total concentration of C minus its initial concentration, there's got to be some algebraic combination, just like I mentioned before, of the molecules that can produce it. So if you start off with A initial, and you know what A is at time t, and you know what D initial is, and D is at time t, then you can calculate the difference between C at time t and C initial. So those are conservation of mass relationships which help you to constrain the solution to your particular situation, because you know what initial concentrations you used. And you have to specify those initial concentrations in the software. So the software, instead of forcing you to do all the math, it does it all for you, whether it's COPACI or virtual cell or even Spring Salad. So let me also discuss another concept called steady state. So at the steady state, all the species, all the molecules in the system have a constant concentration. And for that to be true, the rates of production of those species have to be equal to the rates of their loss, whatever processes those are involved. So all of these rates, R1 and R2, have to be equal to 0 in our particular example. So once you have this kind of situation where the rates are all equal to 0, you're no longer dealing with differential equations. In the previous slide, you know, you had to deal with these differential equations and integrate them. But if all you care about is what happens when everything reaches a steady state, when all the concentrations stop changing, then you can solve algebraic equations. And COPACI can do that for you. That's one of the real strengths of COPACI, and I'm sure you'll hear more about it later. In virtual cell, we don't have that feature yet, although maybe we'll get it one day. We'll probably steal it from COPACI. But you can still do it in virtual cell. All you have to do is start a kinetic simulation and let it run till it reaches the steady state. And then you just copy the final values that are no longer changing. Okay, so you can do that in either software. Of course, you can use that technique in COPACI as well. Here's an important point that I want to make about the steady state. Very often when you're modeling something, you want to see how the system responds to a stimulus, whether it's a signaling molecule, whether it's an uncaging event or a photoactivation, or you want to see how the system changes in response to a stimulus. And you can model that stimulus in any of these software systems. But before you do that, you have to make sure that your system's at steady state. Because if your system is not at steady state when you apply the stimulus, you won't be able to tell what part of the dynamics is associated with the dynamics towards the steady state versus the dynamics that are associated with the response to your stimulus. Okay, so always make sure before you apply your stimulus that you do these things and identify where the steady state of the system is. Finally, before I ask for questions again, I just want to make sure you all realize that you don't have to use those simple mass action laws. Very often when you look at a complex system experimentally, you can't get all those individual rate constants. A good example is Michaelis-Menten enzyme kinetics. Fundamentally, the Michaelis-Menten or enzymes work by mass action rate laws. The molecule A binds to the enzyme E, and that complex then goes into some sort of activated state where A is converted to C while the enzyme is still bound, and then C is released and the enzyme is regenerated. Each one of those steps can be described by mass action, but very often, because some of those steps are so fast, we can't measure those rate constants. And so biochemists, of course, have come up with Michaelis-Menten type approaches to encapsulate all of that information in something that we can observe, the velocity of the reaction and the Km. And so you can put Michaelis-Menten rates into your reaction networks as well. You don't have to do everything in mass action. And for a really extreme example, on the right over here, I'm showing an example of a current going through a membrane that's carried by sodium ions. And it's being controlled by a gated ion channel. And of course, this is the Hodgkin-Huxley model, where Hodgkin and Huxley in their Nobel work did a lot of experiments. They couldn't determine each of the individual steps in this process, but by doing a lot of experiments, they created a phenomenological model called the Hodgkin-Huxley model, which you can see expressed here in virtual cell. Let me just blow it up a little bit here. So here's the Hodgkin-Huxley model, just written out as a rate expression in virtual cell. Okay, so you can write any kind of rate expression you want and allow the molecules to and see how this system behaves with those kinds of kinetics. All right, so that was a lot of information. I expect there might be some questions. I see one hand raised. Gamal, yeah.
Following your description, is this related to the sodium iodine importer that is found in the thyroid?
Say that again, I didn't understand.
The model that you just presented, is it related to the sodium iodine importer that is found in the thyroid gland for the absorption of sodium and iodine?
I'm not sure I know what you're referring to.
Okay, I can send you a direct message so I can put it in right.
Pranathi, I think you were next.
Yeah, hi. I had a more general question actually towards VCell. Does it have like background of machine language and just artificial intelligence in order to produce different models based on like data sets?
So does it have some sort of artificial intelligence, if I understood you correctly, to help you get at some of these parameters and build models?
Yeah, because I thought like with specific data sets and everything, there would be some kind of aspect of machine language to be able to produce like accurate models over time.
No, there's no artificial intelligence in virtual self. All the intelligence has to be the user's intelligence, who can, you know, who actually bases his model on experiments. And if you, one resource that you have in virtual cell is the model database. You can take advantage of all of the very well curated and published models that are collected in our database that are public, and use those as a starting point. So you can search for a starting point for a complicated model. We also can connect to the biomodels database at EBI. That's also a resource available to you.
Okay, I'll go ahead and look into that. Thank you.
Okay.
Questions also on the chat. You should look at that.
Kastub?
Hello, can you hear me?
Yes.
I had a question regarding the stochastic equation. I should have asked this the last time, but I don't think my raised hand was noticed. That aside, you said that the probability of A becoming B is 10% given the energy barrier.
Per second. And 1 second. Yeah.
So does that mean that that particular reaction, what we were talking about, is reversible? That is, B coming back to A is also a possibility. Therefore, the final equation will have a reversible reaction equation.
I'm sorry, you have to ask it again and speak more slowly.
Oh, I'm sorry. The probability of A becoming B is 0.1 per second, right? If
Right.
I remember that. Does that mean that the reaction is reversible? That is, B can become A with a probability 0.9.
For the sake of that example, I didn't allow B to go back to A. But it could go back to A with some other probability. The probability of B going back to A is dependent on the thermodynamics of the system. You know, if let me go back to that slide. Yeah. So if A, so here it looks like they're almost at the same energy. So there might be a possibility that B could go back to A. But if B is very much lower than A in energy, then there's going to be a very, very tiny probability that it goes back to A. So let's just think about it as that, okay? And that has to do with the thermodynamics.
Thank you. I'm sorry I didn't ask it earlier. It's my
That's all
bad.
right. One more question, I think, or no? Gamal, do you have another question? No. Okay, good.
Les,
All right, so let's.
Les, there's questions on the chat.
There's a
There was
question
a
on the
question
chat.
about can a mechanical stimulus be simulated?
Can mechanical stimulus be stimulated? Not directly. We have some experimental approaches to dealing with forces and mechanics that you'll probably hear from tomorrow when Masoud Nakian gives his lecture. We do have some of that in virtual cell, but it's still very preliminary. Even though it's preliminary, it's available to users, but it's only for two-dimensional geometries, not for 3D, and it has certain restrictions. So I'm sure you'll hear more about that from Masoud tomorrow. But direct force type mechanics type stimuli are not possible very generally in virtual cell. Okay.
And there was a final one on the mass conservation, why you did not include B. I think the answer is B is the same as A.
Okay, thank you for answering that, Pedro. Yeah, right. Well, B doesn't have to be the same as A if they had different initial concentrations. So then you would, you could include it, but the difference between B initial and B at time t is going to be the same as the difference between A initial and A at time t.
Yeah,
So,
Exactly.
so those values are going to be the same. Okay. So let's talk a little bit about space. And again, you're going to be hearing a lot more about this from Anne when she gives her tutorial, and then tomorrow from Masood. But let's talk a little bit about it, but not only in terms of doing full reaction diffusion.
Sorry to interrupt. Before you go on with space, there's one participant who couldn't see the screen share, and I just wanted to ask the entire group if there anybody else who experienced problems not seeing the screen share.
I can see it just fine.
Doesn't seem like anybody else has a problem.
Yeah, so it looks like only Jennifer had problems. Maybe Clarissa can get in touch with Jennifer and see what's going on with the Zoom with her.
Okay. All right. So you don't have to solve full reaction diffusion equations to think about space. And in general, I should tell you that as soon as you solve reaction diffusion equations, as soon as you solve them either stochastically or deterministically, you're running into much longer simulation times. So it's always good to run your model first as an ODE type model or a non-spatial stochastic model, what we call a compartmental model, to explore the system behavior. So you can do a lot to consider spatial effects even with what we call compartmental models that only solve ordinary differential equations, not partial differential equations. And these go very, very quickly in any of the software that in either Virtual Cell or Copasi. So let's think about this particular system. This is a very simple system where you have this molecule RANC in one compartment, and it goes through a nuclear membrane. This is the nucleus, goes through the nuclear membrane and goes into the cytosol where you have RANC in the cytosol. Okay, simple flux through the membrane. Let's think about how that system behaves. So it matters what the relative sizes are of those two compartments. And I'm only talking about deterministic solutions. Let's start with 5 micromolar RANC, but this really applies to stochastic as well. We're going to have 0 concentration of RANC in the cytosol. And if the ratio of volumes, if the nucleus is smaller than the cytosol, you'll get this kind of behavior, as you see on the left here. The nucleus will lose a lot more concentration than the cytosol gains. And why is that? Because the cytosol is bigger. If I were giving a class here, I'd ask somebody to explain it, but hard to do that remotely. But just think about it for a second. Obviously, you've got a small nucleus with a certain number of molecules comprising initially 5 micromolar, right? And it's being diluted into a much larger cytosol. So at the end, you're somewhere around 1 micromolar in both compartments, a little bit lower actually. You can just do the math. Again, this is at steady state. You can do the algebra. You don't have to calculate this, but this actually illustrates how you can use the kinetic simulations to arrive at the steady state. Now let's just change the cytosolic volume a little bit. We're going from 15,000 cubic micrometers to 9,000 cubic micrometers, and the steady state goes higher. And of course, if they're at equal volumes, then we end up at 2.5 micromolar at steady state in both compartments, okay? So hopefully everybody kind of intuits that. So space matters, the geometry of these cells matter when you're dealing especially with membrane type processes. Now let's consider one more aspect of this. Let's consider what happens not if I change the volume. I'm going to keep the volumes of these the same, but I'm going to change the area of the nuclear membrane. Remember, the flux was through the nuclear membrane, right? So if I decrease the area of the nuclear membrane, going from an area of whatever it was, I don't remember what I had there, but I decreased it by 75%. So the surface area of the nuclear membrane is lower now than it was before. So there's less area for the molecules to get through. There are fewer channels, total channels for the molecules to get through. You can think of it that way. And therefore, it slows down the kinetics, okay? So that's another way that space matters. But again, you can get the idea of how this works without doing a full spatial model, just taking into account the geometries. All right? So another thing I just want to point out, of course, is that even though the kinetics has changed, the thermodynamics, the equilibrium, is based on the relative volumes of these two compartments. So the final steady state concentration is going to be the same. So even though I changed the kinetics, I haven't changed the thermodynamics, and the steady state concentration still goes to 2.5 micromolar. All right, let's finally consider diffusion in the last couple of slides. This is the full-blown partial differential equation that includes the diffusion. Oh, I forgot to put a D in here. Sorry about that. This whole thing should have parentheses with a D around it. Okay, sorry. So let me, well, I'm not going to draw it, but there should be a D, a diffusion coefficient, that multiplies this entire part of the system, this second derivative of the concentration with respect to the spatial dimensions. Okay, so D, the diffusion coefficient, does that, and that's basically Fick's second law. And all we're doing is adding to Fick's second law a rate expression for how this molecule I changes as a function of time at any given point in space. Okay, this can be reduced to this kind of expression. This sum of these partial derivatives is just the divergence of the flux. The flux is the same flux that you saw in Fick's law in the earlier slides. Okay, and this is the reaction rate. Now this R sub I can be a function not only of C sub I, but it can be a function of all the other concentrations in the system. It can be a function of the voltage across a membrane, if it's a membrane flux reaction, etc. So we're encapsulating a lot of information in this simple term. So you need to consider both diffusion and reaction rates if diffusion is slow on the time scale of these reactions. So for example, for a very, very slow signaling system or metabolic system, you might not need to worry about diffusion because molecules will diffuse very quickly on the time scale of a system that takes, maybe multiple minutes or hours. Most proteins can diffuse throughout the volume of a small cell within a few seconds. But if the process is faster than seconds, or if the cell is very, very big and long, like a neuron, then maybe you do have to worry about diffusion. You also have to worry about diffusion. Another condition is that the distribution of species is not uniform within the cell. So if your starting point is, say, something happening at a membrane, or if a membrane controls the system, then often diffusion does need to be considered because things aren't happening uniformly throughout the entire cell. They're sort of initiating at a membrane. This is what happens during cell signaling, for example. Okay. So very often you do need to worry about diffusion, and that's where a simulator like Virtual Cell can help you. So here are some examples. Here's that same system that I showed you before, where, and you can sort of recognize it from these, from this particular slide. This is just that system where we're allowing the molecule RAN to equilibrate across the nuclear membrane, and eventually it equilibrates to a certain level. And here are the kinetics, okay? You can run this stochastically too, and if you have enough molecules, it behaves pretty much the same as this with a little bit of additional noise, okay? But you can also run it spatially, and here's a cell, okay? And you can see the nucleus over here. You can see the nuclear membrane. This is sort of a 3D rendering of that cell. And I'm looking at the concentration of RAN in the nucleus and in the cytosol, but looking at it at two different points. This point here and this point over here. And if I look at this point that's near the nuclear membrane, I see this kinetics. If I look at it at this point, which is farther away from the nuclear membrane, I see this kinetics. And you can see that both of those are actually kind of different from the kinetics that you see in the well-mixed situation, where we're considering diffusion to be infinite in the ODE situation. So the partial differential equation solution does give you additional information. And likewise, you can also do a stochastic simulation. This is an example now of the average behavior of all of the molecules in a stochastic simulation for the nuclear species and the cytosolic species. And here again, you can see that this is behaving in a different kinetics than you see over here, where the kinetics are a lot slower because you've got diffusion away from the nuclear membrane that's coupled to the actual movement of the molecules through the membrane. Okay, so that is not my last slide, but it's the last part of the formal sort of lesson on reaction kinetics and diffusion. So if we have a few minutes after the questions, I'll go on to a few slides that illustrate some of this in a real model. But let's take some questions first, because that's the priority. Okay, Nitin.
Hi Les, can you hear me?
Yep.
So a couple of questions related to the diffusion model in V-cell environment. So one is related to geometry of the problem. So when you simulate different shapes, does the V-cell adjust the coordinate systems from a rectangular to cylindrical or other coordinate systems?
You can put in any geometry you want, basically, and you'll see that in Anne's tutorial.
And other is related to boundary conditions. So the users define the boundary conditions at the specific interfaces, or the vCell has a particular set of boundary conditions that it takes in that model?
The only place that the user has to define the boundary conditions is at the end of the edges of the simulation space. You don't have to define the boundary conditions at membranes within the model. The software calculates that automatically, depending on the fluxes and the on-off rates of molecules moving on and off the membrane, et cetera, et cetera. But at the edges of the world, at the edges of the environment, of the simulation container, that's where you do define boundary conditions. So you can define a boundary condition, for example, as being closed, that is a zero flux boundary condition. You can define a boundary condition at the edge of the world as being some expression in terms of fluxes at those areas. You can also define what we call value boundary conditions, where you fix a certain value of some species, let's say in the extracellular space. Let's say you have some signaling molecule that's approaching, that needs to bind to a cell surface receptor. That signaling molecule is on the outside of the cell. You've got that cell in a dish with an essentially infinite supply of that molecule. So you can fix the value of that signaling molecule to whatever concentration is in the dish at the edge of the boundary. So you can do all that stuff in Virtual Cell. Okay.
Okay, thank you.
Shraddha?
Hello, Les. Shraddha here. My questions might be a little naive because these concepts are very unfamiliar to me. This was regarding diffusion. So if you have a cytosolic compartment and you have maybe 2 or 3 nuclei of the same volume, would steady state be reached at the same time?
If you had like 2 nuclei at the same volume, and you had, let's say, this RAN-C at that initial concentration in both the nuclei, and if you did a compartmental model without worrying about diffusion, then the answer is yes.
Okay.
Steady state would be reached at the same time. If you had some sort of geometry that wasn't that symmetrical or something, then steady state would not be reached at the same time.
Sure. And when do you know if you need to run a stochastic calculation or a deterministic calculation? Do you just run both, or it's dependent on the number of molecules that are there?
Usually, you are limited in terms of how long you want to wait for your simulation to end. So
Sure.
Stochastic simulations are more computationally intensive. For non-spatial, it's not a big deal. You could run a stochastic simulation with many molecules if you want to. But in general, you're not going to get a very different answer than you would with deterministic if you have many, many molecules. So you could, try a stochastic simulation and see if the behavior is fundamentally different. And there are some really exciting papers, but I think I can count them on one hand, where a stochastic simulation gives a completely different result than than a deterministic simulation. But very often you can get more insight into the deterministic behavior if you run a stochastic simulation. There's going to be a complete lecture tomorrow by Pedro on stochastic simulations.
Okay, thanks for that.
Yeah, we will see some examples of cases when it's not the same result.
Hania?
Yes, so I have a question. So the simulation and result that you show, it's just the 3D diffusion inside the cytoplasm or the nucleus. Is it correct?
Yes.
And if I want to do some diffusion on a membrane, even in the just a cell membrane or the nucleus membrane, is it possible to do that in this package?
Yes, we have surface diffusion enabled. It's done basically in the same way. You just, yeah, yes, the answer is yes.
And just more question related to this topic. So you mentioned that the geometry of the nucleus or just the geometry is important for the diffusion. So my question is behind this model of behind the diffusion equation in this package. So for example, do we consider the geometry of the shape? I mean that, for example, if the curvature is high or low, if the diffusion is changing or not, something like that, because I am interested, for example, to test if the geometry, that the curvature of the geometry is important for the diffusion constant.
Yeah, again, when we have our discussion tomorrow of a more deep discussion by Masood on spatial simulations, he'll cover all that. But the bottom line is that if you want to model diffusion where the geometry has very fine features, very tiny features, then you have to use a very fine simulation mesh. And those simulations can be long, depending on how complex the reactions are, etc. But yes, the answer is yes, you can do that.
And it's in the virtual cell package.
Correct.
Thank you so much.
Gamal, you have one last question? Yes, very quickly. On your last image for the single cell that you show, you use voxels to do the simulation. Am I correct? Are these voxels isotropic?
Yes, they're isotropic in each dimension. So in other words, you can have different sizes, different dimensions in X compared to Y, but within X, they all have to have the same dimension.
Correct. So very quickly, so you can set up the voxel size to speed up the calculation, bigger, smaller.
Right, but as I mentioned in the last question, you have to make sure that the size of the mesh, the size of the voxels, the size of the grid is fine enough to capture all of the geometrical features in the actual geometry.
Mm-hmm. So,
okay.
There's also the possibility to use a variable mesh size using a specialized solver called EV-Jumbo.
Yeah, but that's also very experimental, and generally we found that it's slower than what we get with the default solver.
So can you tell us very quickly what is the voxel size that you use on the specific model? Is it about 0.1 micron, 0.01 micron?
So, I use that model for the tutorial that I'm going to just spend the next hour on. So we'll get to discussion of mesh sizes there. If you want to hold on to that, you'll see how to set it.
I think we're at running into Anne's time, so I'm going to stop at this point. I'll just say that I was going to talk about a paper that illustrates all of these ideas. It's an old paper, but it's still a very powerful use of some of these ideas. And I put it into the Google Drive so you can all look at that. And it's also in the PowerPoint. That's also in the Google Drive. So, you know, all of these we'll have to skip. Okay, and I'm going to turn it over to you.